Hello, welcome to the second part of our lecture on functional neuroanatomy. Last time we discussed the surface features of the brain. We talked about how to name and how to understand the directions of the brain, looking at it from the outside. We talked about the cerebral cortex, which is the most prominent feature from looking from the outside of an adult human brain. We talked about the four lobes, and we also talked about how the cortex is crossed with respect to the functions that it serves. So, for example, the right side of the brain deals with the left side of the body, and vice versa. And we finished up by talking about the planes of section, these slices that we take through the brain in order to visualize its interior structures. This is where we're going to pick up, and we're going to talk about the sorts of things that we see using these planes of section. We're going to talk about specifically how to identify gray matter and white matter and the different kinds of gray and white matter that we see. Then we're going to talk about the major divisions of the central nervous system. There are several different divisions that we'll get into, and we're going to focus on two of those, the spinal cord, and we're going to go into even greater depth into the cerebral cortex. All right, so you'll remember we have three sort of main planes of section. There are these imaginary slices that we take through the brain, and they come at right angles to one another. The first one we talked about last time was the sagittal section, then the horizontal section. This is also called an axial section, and finally the coronal section. And what we notice when we take these slices through the brain is that we begin to see the sort of organization and the structure of the interior of the brain, and we even get to appreciate the degree to which the cerebral cortex on the outside is so deeply folded and complex. Now, what is it exactly that we're seeing when we see these, for example, these differences in color here? Well, what we're looking at here is gray matter and white matter. And to understand what they are, we have to go back and remind ourselves that the brain, of course, is made of cells, and the cells of the brain, the neurons of the brain, in general have two main parts. They have their cell bodies and the axons. And throughout the brain, the cell bodies tend to cluster together, and when they cluster together, whether it's on the cortex, when they tend to cluster together on the outside of the brain, it tends to form the cortex, which we've talked about. They also happen to cluster together on the inside of the brain, and they form what are called nuclei. So both nuclei and both the cortex contain what are called gray matter. So when you hear gray matter, think the cell bodies of neurons in the brain. The axons of neurons also tend to cluster together or run together, and when they do that, we collectively call them white matter. Now, when axons form white matter inside the brain, we say that they form tracts within the brain. When they do that outside of the body, we say that they form nerves. So whenever you think of white matter, think of the axons all running together in a bundle. Now, the words gray matter and white matter can be a bit confusing because depending on how you're looking at the brain, the gray matter isn't always going to be gray, and the white matter isn't always going to be white. So what I'm going to do is just show you a couple of different ways of visualizing the brain, so you can just get used to, identifying gray and white matter through different imaging methods. The one that might be most familiar to you, and especially if any of you are going to go into the medical field, is the MRI, magnetic resonance imaging. This is mostly what people talk about when we say a brain scan. But MRIs come in different flavors. The one on the left is what's called a T2 weighted MRI, and the one on the right is what's called a T1 weighted MRI. And you can see, the general shape of things here is similar, although the color scheme, the gray and white scheme, is totally reversed. In a T2 weighted MRI, anything that's filled with fluid, like these ventricles here, is going to stand out as bright white, but in T1 weighted, fluid is going to be dark black. In a T1 weighted image, the white matter that you see here, and you can see the corpus callosum here as an example of a big white matter tract, the white matter is always going to be fairly light in color, whereas white matter is going to be much darker in color in a T2 weighted MRI. And for both T2 and T1, the gray matter, you can see the gray matter of the cortex here and here, is going to be somewhere in between. It's not going to be as bright. It's not going to be the same color as the fluid-filled spaces, nor is it going to be the same color as the white matter spaces. It's going to be in between. So you may, in your future career, see either T2 or T1 weighted MRIs, and you should be able to recognize gray matter and white matter and fluid-filled spaces like the ventricles in both of them. Another way of visualizing the brain can be done in what are called fixed sections. So this is an instance where you take a whole brain specimen and you preserve it, slice it into very, very thin slices, and then impregnate that slice with some sort of dye, or you treat it with some sort of chemical that will make different features stand out. So on the left here, we have a section of a human brain that was labeled with a cell body stain. So of course, it's going to label the cell bodies, the gray matter, much darker than the white matter. This particular cell body stain, I believe this is a Nissl stain, has the extra property of, if you zoom in to the cerebral cortex, letting you see the layered structure of the cortex. And we're going to get into the cortical layers a little bit later. Over here, on the other hand, this is an axon stain. So anywhere that there are axons, it's going to stain this sort of dark cocoa brown. So you can see, if you zoom in here, you can see this part here, this pale area that isn't labeled at all, is where all of the cortical gray matter is, where all of the cell bodies are. And just looking at these two side by side, you can hopefully appreciate that they're complements of one another. Now, if you look here in this section with the axon stain, what you can see is that certainly there are these big pale regions on the outside, but there are also these pale regions on the inside. So what does that tell us? Well, if it's not made up of axons, and it's not a ventricle, then what it tells us is that this must be gray matter. These are two gray matter nuclei that are beneath the surface of the cortex and located deep in the brain, and we can identify them on the basis of the fact that they are not being stained with this axon stain. And you can see them here in the cell body stain too, although to my eye at least they seem to stand out much more over here. All right, now what I'm going to show you now is a really fun movie that I found, and I'll make sure to post it on Blackboard. So this is a little sort of journey through a human brain done in coronal section. And what we're going to do is we're going to take a movie. We're going to start our coronal section up here, taking a slice off of the very anterior part of the frontal lobe, and then we're going to sweep back. And as we do, I'm just going to narrate here some of the things that we see. And we're doing this just to hopefully kind of give you a more complete picture of how all of these parts fit together. Now this is a T2 weighted MRI. So the cell bodies, the gray matter, are going to take on this light gray color. The axons are going to take on this dark gray color. The one trick here is that the ventricles are also going to be dark black. They're not going to be bright white like in this T2 MRI, and that's because this T2 MRI that I'm circling right here was taken from a living human brain, and so the ventricles are filled with fluid, as they are in your brain and mine. This was taken from an ex vivo specimen, so this is a brain that was taken out, that was preserved, and was scanned at high resolution with an MRI. There's no fluid in these ventricles, so they're not going to show up bright white. Just don't want that to throw you. Okay, oh no, let's play this video. There we go. Okay, so you see we're beginning our journey here. We're right now at this moment just in the tip of the frontal lobe here, and you can see very clearly defined white matter and gray matter. You can see the two hemispheres very clearly defined. And in just a moment, we're going to see two interesting features, two features that you should already be able to identify. All right, and let's go to the right part of the video. Okay, my apologies. We're going to try this video one more time. And we're going to pick up right about here. So there should already be a couple of features that you should be able to recognize. Remember, this is the white matter, this darker substance here, and we see the white matter now is connecting the two hemispheres, so you should be able to name this as the corpus callosum. And these spaces here that are sort of below and to either side, you should be able to recognize as the ventricles. And we're going to keep advancing through. We're going to keep going a little bit, and I think here's a good place to stop. All right, first of all, we'll see that in the ventral portion of the cerebral cortex here, we notice now that it's sort of split off. It used to just be two hemispheres and then two hemispheres joined by the corpus callosum, but now we have these sort of extra lobules down here. And you should be able to recognize these as the temporal lobe. This is the very tip of the temporal lobe that we're now slicing into as we go from anterior to posterior. Hopefully you also notice now in the deep within the brain these two pale regions here, and because of course they are these pale regions located deep within the brain, and they're not part of the white matter, they must be the gray matter. And what you're seeing now here are the gray matter of a structure called the basal ganglia. And the basal ganglia is a set of several different nuclei. These two nuclei here are called the caudate and the putamen. And you'll see, in fact, that the caudate and putamen, at least at this level, are separated by this band of white matter on either side. This band is called the internal capsule. And the internal capsule consists, of course, of axons, but axons that are destined for a very particular purpose. These are axons whose cell bodies are up here in the cerebral cortex, originating in the cerebral cortex, and are projecting down through the brain, and are eventually going to be projecting down into the brainstem and into the spinal cord. So some of these are going to be the axons of neurons that are directly synapsing on motor neurons in the spinal cord and controlling movement. Okay, let's resume. And you'll see now that the temporal lobe has joined up with the rest of the brain here. So now we're taking a slice about midway through, and we're seeing that the temporal lobe is joined up. And what you can also see here, remember that this fissure here, this lateral fissure here and lateral fissure here, is what separates the temporal lobe dorsally and medially from the frontal lobe and the parietal lobe. And we can see that this lateral fissure goes very, very, very deep. There are these folds of the cortex that are hidden deep, deep within, and this is called the insula. And it's something that we wouldn't see from the outside, but we can see very easily here in this coronal section. We're continuing through. We can see the gray matter here of these axons projecting down. Now, right now, what we're seeing here is a part of the hindbrain called the pons, which we'll be talking about in just a moment. These large spaces here are continuances of the ventricles, and we'll stop here. Now you'll see this very different change in texture, if you will. You'll see that the gray matter here is much denser and tighter and has this different branching shape. Now we're at the back of the brain here, so what must this be? This has got to be the cerebellum. The cerebellum has this very unique, characteristic pattern to its gray matter that's very, very different from the gray matter of the cerebral cortex. And we're going to let this Continue on, you'll see our ventricles are just starting to vanish. We're seeing now deep within the cerebellum what are called these dentate nuclei. These are, gray matter nuclei that are inside of the white matter of the cerebellum, which is itself inside the outer cortical area, the gray matter. The cerebellum, the Latin name means little brain, and you can see how it's a lot like the larger part of the cerebral cortex, right? The cerebral cortex, if you remember back here, we have gray matter on the outside, white matter interior to that, and then gray matter within that. And the same is true of the cerebellum. Gray matter, white matter, and then gray matter again. All right. I'm going to post this video. You can look through it yourself. And this channel also has a couple of other videos like this. All right. So we've talked about gray and white matter, and we sort of navigated our way around. We're going to talk now about some of the major divisions of the central nervous system. Before we get to that, I just want to take a pause here. So when we talk about the functions of the brain, what sort of functions are we talking about? What does the brain do for us? So first of all, the brain generates our movements and other behaviors. It processes sensory information and constructs the sensory reality that we experience every day. The brain is responsible for forming our memories, for allowing us to learn from past experience. And in addition to that, the brain helps regulate our basic bodily functions, breathing and digestion and those sorts of things. So when we talk about functional anatomy, what we're trying to do is figure out how the brain contributes to one or more of these basic functions. So when we're thinking about a part of the brain, let's sort of always try to think back to these basic functions that we have, whether it's behavior or regulation of the body or memory. Now, the central nervous system in mammals is divided into these sections. We start all the way at the very bottom with the spinal cord. Moving up, we come to the myencephalon. Next, we have the metencephalon, which includes the pons and cerebellum, mesencephalon, which is sometimes called the midbrain, diencephalon, and telencephalon, which includes the cerebral cortex, the most complicated and developed part of the mature human brain. Now, why do we divide them up like this? Where do we get these names? These divisions are based upon the fact that basically all mammals and most vertebrates sort of all share the same basic architecture for their brains. So in other words, we can take a fully developed human brain, we can compare it to a mammalian embryo, and we can match these parts up one to one. And we can even take a mammalian brain and compare it to some lower vertebrate, like a reptile or something like that, and we can find parts of the brain that match up to it on a more or less one-to-one basis. And these are in fact these parts of the human brain here, of the developed mammalian brain, are ones that evolved from simpler structures that are present in current day species like reptiles and things like that. So these names and this organization wasn't just totally made up. It comes from a very evolutionarily motivated place. It allows us to sort of compare the anatomy across species and even in vastly different species. Now, let's talk about some of the functions. And you remember back when I talked about the cerebral cortex, and I said when we talk about the four lobes of the cortex, it's kind of like talking about area codes. Or I'm sorry, it's kind of like talking about time zones, because within a time zone there are many different subdivisions. The same really is, and that's true for the cortex, but it's true really for all of these. So, for example, starting up here with the diencephalon, the diencephalon contains a number of different structures, including the thalamus, the hypothalamus, and the pituitary gland. And these all have different functions. The thalamus, for example, is responsible for the routing of different kinds of sensory information. The hypothalamus is important for regulating some of our basic drives, like eating and sleeping. And the pituitary gland regulates some of our functions related to hormones. If we go down a level here to the mesencephalon, this is called the midbrain. This contains structures that govern what are called orienting movements. Orienting means sort of moving the eyes or the head or the body in order to look at something. You know, if you hear a sudden sound or see some movement out of the corner of your eye, your eyes and your head and your body will automatically move and orient towards it. A lot of those movements are governed by structures in the midbrain here. But in addition to that, the midbrain also contains a large number of dopamine neurons, and dopamine neurons have functions. Dopamine has functions all throughout the brain, and many, many different functions. So again, this is a part of the brain that contains many different subparts with different functions. Below that, we have the metencephalon. This includes the cerebellum and the pons as well. The cerebellum, we spoke of it before, is responsible for balance and coordination of complex movements. And some of that is also governed by the pons as well. The pons basically contains a lot of inputs and output structures that connect the cerebellum to the rest of the brain. Moving below that, we have the myencephalon. Here we're getting into a part of the brain that governs many of our most basic functions, in particular the control of breathing and the control of our heartbeat. And of course, the spinal cord governs our motor inputs and outputs. Now, look, I know this is a lot of very general information. If you want some more specifics, you can definitely go read the textbook. The one general, maybe sort of governing principle that you can take away from this is that as we go from spinal cord to myencephalon to metencephalon and on up, we get to gradually more complex neural functions, right? We start down here at the spinal cord, which deals with sensation and movement, and up from that, we have this part of the brain which contains the medulla, the breathing center, and as we go up, things generally get more complex, all the way up to the cerebral cortex, of course, which governs all of our most complex behaviors and cognitive activities. All right, now what we're going to do for the last part is focus just on two of these sections. We're going to talk a little bit more about the spinal cord in depth and a little bit more about the cortex in depth. So this here is a section through the spinal cord. So if you imagine just laying the spinal cord out on a table and just chopping it right down the middle. This is a cell body stain. So that tells us that this must be gray matter, and all of this around here on the outside must be white matter. And we see that in the spinal cord, we generally divide it into a dorsal horn and a ventral horn. And remember, we call this the dorsal part of the spinal cord because the human neuraxis is bent. If you as a human were to get onto the ground and get on all fours, this part of your spinal cord would be facing up in the dorsal position. This part of your spinal cord would be facing down in the ventral position. Now, what about the neurons here in the gray matter? Well, the spinal cord is sort of very, very nicely organized. The dorsal part of the spinal cord generally contains sensory functions. So in other words, we can see in this illustration here a sensory neuron, a primary sensory neuron that is detecting inputs coming from a stretch receptor in the quadriceps muscle, and it's forming its synapses here in the dorsal horn. In the ventral horn of the spinal cord is where most of the motor neurons lie, where most of the neurons that eventually go back out and synapse onto the muscles live. Now occasionally, these neurons directly connect to one another. So you can see this neuron, this sensory neuron in this illustration, is directly connecting to this motor neuron, and it's a circuit like this which governs some of our most basic reflex movements that really don't need any sort of input from the brain. And one thing to keep in mind, or one thing to know, is that this sort of basic unit here, this basic sort of input-output unit, is repeated all throughout the spine. And for every segment of the spinal cord, there's a different region of the body that is served by it. So you can see, for example, on this map, These are your cervical vertebrae and cervical nerves governing your head and your shoulders and your upper arms. Thoracic nerves govern the trunk, lumbar nerves about the lower trunk and the front of the legs, and the sacral nerves, the posterior and the lower part of the legs. This, if you're going into the medical field, is going to be very important information, right? Because if somebody comes in with some injury to their spine or injury to their back and they're experiencing some form of paralysis or loss of sensation, depending upon where that paralysis is, you should be able to locate the location of the injury. And the reason, of course, there's paralysis is because the spinal cord just doesn't contain the gray matter here. It contains all of this white matter, all of these axons that are connecting the gray matter to the brain above it. So if there is a transection or an injury or a crush in part of the spinal cord, you're not only interfering just with the gray matter that happens to be located there, you're interfering with the communication that occurs between all of this part of the spinal cord and the brain. All right. We're going to return to the cortex now. This is going to be our last sort of little bit of focus. And you'll remember, I told you before that when we talk about the human cerebral cortex, what we're talking about is neocortex. And what does neocortex mean? It means that it is 6-layered cortex with 6 layered. There are other forms of cortex. For example, allocortex describes cortex that has either 3 or 4 layers, structures like the hippocampus or the piriform cortex. And humans have allocortex too, as do sort of non-mammal species like birds and reptiles. It's just, in humans, most of our cortex is the 6-layer neocortex. Now, the interesting thing about the cortical layers is that if you look closely at them, you can, first of all, sort of see the same general organization throughout the whole brain, but the organization there, or the exact patterning of the layers, differs from one region to the next. So, for example, layer 4 of the cortex, and this is true of the cortex throughout the brain, layer 4 of the cortex generally receives inputs. Layer 5 of the cortex generally contains cell bodies that send outputs to other regions. Now, they show you a little bit of a comparison between motor cortex and sensory cortex here. I'm going to show you a bit better of an example. This is an illustration of the organization and the sort of the packing of cells in motor cortex. Here is the same thing for sensory cortex. Now, it takes a bit of a trained eye to recognize that this is layer 4 in the motor cortex and this is layer 4 in the sensory cortex. But what you can at least be able to appreciate is that layer 4 here in the sensory cortex is much, much more developed than layer 4 here in the motor cortex. So what that tells you, what that gives you a clue to, is the fact that this cortex here is going to have much more of a function processing sensory inputs than this cortex here with the minimal layer 4. Now, the sort of study of the cortical layers here is called cytoarchitectonics, and one of the sort of most famous illustrations in neuroscience is what's called the cytoarchitectonic map of the human brain. And I'm showing you an example of this sort of map here. So this is a map, a very old map that was first created in 1909. This here contains a map of different regions of the cortex, and what makes them different is that the layering of the cells in each of these regions is subtly different from one another. And you'll remember back, in the first lecture, I said that, the different lobes of the brain are kind of like the time zones, and that each, just like the time zones, each lobe is subdivided into smaller regions. Well, here now, we're seeing a map of all these different regions. Now, in case this is hard to visualize, I've also shown an example of a more modern map here, sort of developed with more modern imaging techniques, but it's really sort of showing us the same thing. And the sort of take-home here is that we can, divide up the cortex in a fairly fine-grained way into different regions based solely on the structure of the cells that are there. And in addition to that, we can ascribe, functions to these different regions, um, based upon their inputs and on their outputs, and based upon some other techniques. And I'm just listing some of them here. In red here, we've highlighted the primary somatosensory cortex. So this is processing our bodily sensations. Visual cortex in the occipital lobe here, primary visual cortex. This is the first part in the cortex that processes visual input and helps with our sense of vision. For the auditory sensation, it's this area here in orange, area 42. Area 4, which is this area with these sort of big black dots. This is our primary motor cortex, just on the other side of the central cortex. This is the part of the brain most responsible for controlling voluntary movement. Now, beyond these sort of primary sensory motor areas, there are also some other areas that perform more complex functions, and I've just highlighted a couple of these here. You might have heard of Broca's area, which I've put in the green circle here. Broca's area has to do with the production of speech. And then sometimes you'll hear about this area, including areas 9 and 46, called the dorsolateral prefrontal cortex. This is the part of the frontal lobe that deals with reasoning, planning, and working memory. Okay, so to sum up what we've talked about today and some of our learning objectives for today, we talked about the different planes of section and about how to identify gray in white matter in MRIs or in stained sections. And hopefully by now you should be able to find some of the major features: the neocortex, the ventricles, the corpus callosum, the cerebellum, and subcortical gray matter structures. We talked about the major divisions of the mammalian central nervous system, and by the end of this, you should be able to name at least one major function for each of these, and to be able to identify them on a drawing. And then we talked a little bit more in depth about the function of the spinal cord. For the spinal cord, you should be able to identify where the gray and white matter are, where the sensory and the motor neurons are, and to be able to describe how a simple reflex circuit might work. And finally, we talked about the cortex, specifically difference between neocortex and allocortex, and you should be able to, again, identify the 4 lobes, and you should be able to, in addition to identifying the 4 lobes, you should be able to identify and understand the function of at least 1 or 2 of the more specialized cortical areas that I described here. All right, that's the end of our functional neuroanatomy lecture. The next topic will be on the functions of neurons. I look forward to it. Have a good day.